continuation with the previous video where I discussed regarding the structure of human digestive system. In this video, we'll talk about the process of digestion. You all know that digestion is a process where the complex food particle that is ingested into the mouth or the oral cavity is broken down into simpler soluble form, and whatever the undigested or unabsorbed waste will be thrown out of the body in the form of feces. So that is the definition of digestion. But we'll study today what are the process that takes place during the digestion, especially in different parts of the alimentary canal. The process of digestion is accomplished by mainly two processes, that is mechanical process and the chemical process. So different types of mechanical and chemical process takes place in different parts of the alimentary canal. Anyhow, this mechanical and the chemical process should start with the buccal cavity, because the food particle is first ingested into the buccal cavity. That's why the process of digestion should also start in the buccal cavity. And in the buccal cavity, mainly two types of work is done. That is mastication of the food and facilitation of the swallowing. So these two are the important step that takes place in the buccal cavity. What do you mean by mastication of the food? Mastication means grinding, and you all know that grinding of the food or the mastication of the food is done with the help of teeth and the tongue along with the help of saliva. So saliva is secreted by the salivary gland that are present in the mouth region, and it secretes saliva into the buccal cavity. So teeth and the tongue with the help of saliva will grind the food particle or masticate the food particle, during which the food particle get thoroughly mixed with the saliva which are secreted by the salivary gland. So mixing or the grinding or the mastication of the food is nothing but the mechanical process. And what about the chemical process? Yes, chemical process of digestion also takes place in the mouth cavity or the buccal cavity, and that is done with the help of content of the saliva. Saliva mainly consisting of electrolytes like sodium, potassium, chlorine, bicarbonate, etc. Apart from that, saliva also consisting of enzyme known as salivary amylase, lysozymes, etc. And these enzymes helps in the chemical process of digestion in the buccal cavity. First one is salivary amylase. Salivary amylase is a hydrolytic enzyme. Hydrolytic means it will break something into simpler form. It breaks what? It breaks a complex carbohydrates like polysaccharide, example starch. So these salivary amylase means one which will break down the carbohydrate or carbohydrate splitting enzyme. So salivary amylase, it will break complex carbohydrate or polysaccharides like starch into disaccharides like maltose. So salivary amylase, it will split starch into maltose at pH 6.8. So saliva in the buccal cavity provide that optimum pH for working of the salivary amylase, which will break down complex carbohydrate like starch into the maltose. So in the buccal cavity, starch is converted into simpler form, that is disaccharide, that is maltose, with the help of salivary amylase. That is the first enzyme. Another enzyme is lysozyme. Lysozyme won't split any food particle. Instead, it acts as an antibacterial agent, because through the food particle many microorganisms or infectious microorganisms can enter the body, and that will be tackled by this enzyme, that is lysozyme, which act as an antibacterial agent. So that is how mechanical and chemical process of digestion takes place in the buccal cavity. So mechanical process by mastication or grinding of the food particle, and chemical process by salivary amylase which act on the starch and convert it into maltose. So during this process, the saliva thoroughly get mixed with the food particle. Apart from that, saliva also consisting of mucus, and this mucus helps in lubrication, as well as it helps in the attachment of this masticated food together to form a bolus. Bolus is a mass or the ball of this masticated food particle. So mucus which are present in the saliva helps in attachment of this masticated food particle to form a bolus. Later, this bolus is conveyed into the next part, that is pharynx, by swallowing or deglutition. So, by swallowing or deglutition, the bolus, that is the ball of the masticated food particle, is conveyed into the next part, that is pharynx. Later from the pharynx it is conveyed into the esophagus. So esophagus is a very long muscular tube, and in the esophagus this masticated food particle, that is the bolus, passes through this esophagus by the series of muscular contraction of walls of the esophagus. So there is a series of muscular contraction of the wall of the esophagus, and that contraction is known as a peristalsis. So through this peristalsis movement, the bolus passes down the esophagus and finally it enters the stomach. Anyhow, in between the stomach and esophagus, there is a sphincter, that is gastroesophageal sphincter, which will control the entry of food from esophagus to stomach, and it prevents the backflow of food from stomach to esophagus. Now, the food is present in the stomach, and you all know that the wall of the stomach, or the inner lining, that is mucosal layer of the stomach, consisting of gastric gland, and these gastric gland consisting of mainly three types of cell, or it is made up of mainly three types of cell, that is mucous neck cell. As the name indicates, this mucous neck cell secretes mucus. Second type is peptic cells or chief cells. These peptic cells or chief cells secretes proenzyme pepsinogen. Proenzyme means this enzyme is in the inactive form. So proenzyme pepsinogen means this pepsinogen is in the inactive form, that is secreted by peptic cell or the chief cell. Third type of cell is parietal cell or the oxyntic cell, and it secretes HCl. Yes, it secretes hydrochloric acid. So acidic condition of the stomach is because of the presence of this HCl or hydrochloric acid, which is secreted by parietal cell or the oxyntic cell, and it also produces an intrinsic factor which helps in the absorption of vitamin B12. So these are the secretion of different types of cells of the gastric gland, which is present on the mucosal layer of the stomach. Now we'll see how these secretion helps in the digestion of the food particle. So as I already told, now the food is present in the stomach and food remains in the stomach for 4 to 5 hours, during which these food thoroughly mixes with the content of the gastric gland, or it thoroughly mixes with the acidic gastric juice. Gastric juice is a juice that is produced by the gastric gland or content of the gastric gland. Why it is known as acidic gastric juice? Because it also secretes hydrochloric acid. Is an acid. That's why it is known as acidic gastric juice. So the content, that is masticated food or the bolus, got thoroughly mixed with the acidic gastric juice by the churning movement of the muscular wall of the stomach. Churning movement is nothing but the contraction and relaxation, series of contraction and relaxation of the walls of the stomach, or churning movement of the muscular wall of the stomach, with the help of which the food got thoroughly mixed with the acidic gastric juice to form a chyme. So chyme is nothing but the food that is got thoroughly mixed with the acidic gastric juice. So whatever the food particle that is present in the stomach is known as a chyme. So upon mixing of the food particle or the masticated food thoroughly with the acidic gastric juice in the stomach, the pepsinogen, that is in the inactivate form, got exposed to the hydrochloric acid, that is acids of the gastric gland. So upon exposure of this inactive, that is proenzyme pepsinogen, to hydrochloric acid, it converted into its active form, that is pepsin. So upon exposure of a pepsinogen, that is inactive form, into the hydrochloric acid, it is converted into its active form, that is pepsin. So hydrochloric acid is required for the conversion of inactive pepsinogen to active form, that is pepsin. And this pepsin is a proteolytic enzyme. What do you mean by proteolytic? Prote means protein, lytic means splitting, so it is a protein splitting enzyme. So pepsin is a proteolytic enzyme of the stomach which splits protein into its simpler form, that is proteoses and peptones. So proteins are converted into proteoses and peptones in the presence of pepsin, at the pH 1.8, that is acidic pH, which is maintained by the presence of hydrochloric acid. So in the stomach, this acidic pH is maintained, which helps in the conversion of protein into proteoses and peptones in the presence of enzyme known as pepsin. So that is the function of pepsin, which converts most of the protein into simpler form, that is proteoses and peptones. Apart from that, gastric gland also produces certain amount of lipases. So lipases means which splits the lipids. So it helps in the digestion of lipids also. In the infants, they also have another proteolytic enzyme known as renin. So renin is a proteolytic enzyme which are present in the infants that helps in the digestion of milk protein, because they take only milk, mother milk. So digestion of the milk is done with the help of an enzyme, that is proteolytic enzyme known as renin, which will splits the milk protein. So that is only present in the infants. Whereas in the adult, they consisting of pepsin in the inactive form known as pepsinogen. Upon exposure to HCl, it is converted into pepsin, and which helps in the digestion of proteins into proteoses and peptones. So that is how most of the proteins in the stomach is converted into its simpler form, that is proteoses and peptones. Apart from that, gastric gland also produces mucus and bicarbonate, which helps or which will protect the lining of the stomach, because the content of the stomach is hydrochloric acid. If the hydrochloric acid comes in contact with the wall of the stomach, it leads to inflammation or the irritation. So to prevent that, the mucus and the bicarbonate is produced by the gastric gland. So mucus forms a layer just above this mucosa layer or inner lining, and even the bicarbonate. So bicarbonate is a base, isn't it? So this base or the bicarbonate neutralizes the acid which is present near the wall of the stomach. So mucus and bicarbonate thereby protects the inner lining of the stomach from this corrosive hydrochloric acid. So that is what happens in the stomach. So mainly proteins and the lipids are converted into its simpler form, but not carbohydrate. Yes, digestion of carbohydrate does not take place in the stomach because of its acidic condition. The digestion of the carbohydrate is paused in the stomach. Only digestion of proteins and lipids takes place in the stomach. So that is about the digestion in the stomach. Food, after staying in the stomach for 4 to 5 hours, next it enters the small intestine. Specifically, it enters the duodenum part of the small intestine, where it gets mixed with the secretion of the pancreas known as pancreatic juice, secretion of the liver known as bile juice, and secretion of the small intestine known as intestinal juice. So food in the duodenum got thoroughly mixed with the pancreatic juice, bile juice and the intestinal juice by the muscular movement of the small intestine. So upon various types of movement shown by the muscular wall of the small intestine, food thoroughly mixed with the pancreatic juice, bile juice and the intestinal juice. And these juices consisting of different types of content or the enzyme which helps in the digestion of the food that is present in the duodenum. So first we'll see the content of these juices. First one is pancreatic juice, which is secreted by the pancreas, and this juice consisting of enzyme in the inactive forms like trypsinogen, chymotrypsinogen, procarboxypeptidase, amylases, lipases, nucleases, etc. So trypsinogen, chymotrypsinogen and procarboxypeptidases helps in the digestion of protein. Amylases, as the name indicates, it helps in the digestion of carbohydrate. Lipases digest lipids. Nucleases helps in the digestion of nucleic acids. So these are in the form of inactive form. First it should be get activated. So trypsinogen first get activated by another enzyme known as enterokinases, which is secreted by the intestinal juices. So enterokinase is secreted by the intestinal juices, and this enterokinase in turn activates the trypsinogen into its active form, that is trypsin. Later this trypsin activates the other inactive enzyme. Apart from these enzymes, pancreatic juice also produces mucus and bicarbonate. So this mucus forms a lining of the duodenum or lining of the small intestine, because the content of the stomach is acidic content. So if this acidic content enter the duodenum, it may irritate or the inflammation may occur in the wall of the duodenum. So to protect the walls of the duodenum, the mucus which is secreted by the pancreas forms a layers on the duodenum wall. Apart from that, the bicarbonate which is secreted by the pancreatic juice neutralizes the pH, because the pH of the stomach is acidic pH. So upon entering into the duodenum, the bicarbonate neutralizes this acidic condition into alkaline. So pH of the small intestine is alkaline pH, that is 7.8. So pH of the buccal cavity is neutral, that is 6.8. The pH of the stomach is acidic, that is 1.8. And pH of the small intestine is alkaline, that is 7.8. It is done with the help of bicarbonate which is produced by the pancreas or which is present in the pancreatic juice. So these are the secretion of pancreas or the content that is present in the pancreatic juice. Next one is bile juice. So bile juice is produced by the liver and it is temporarily stored and concentrated in the gallbladder, and it enters the duodenum through the hepatopancreatic duct. So pancreatic juice and the bile juice is secreted into the duodenum through the hepatopancreatic duct. So this bile juice consisting of pigments, that is bile pigment known as bilirubin and biliverdin. So bilirubin and biliverdin are the pigments, that is color, which gives a color to the feces. So bile pigment, that is bilirubin and biliverdin. Apart from that, it also consisting of bile salts, cholesterol, phospholipids, etc. But bile juice lack enzyme or no enzyme is present in the bile juice. Then what is the function of bile juice? Bile juice, that is content of the bile juice, helps in emulsification of the fat. What do you mean by emulsification? Emulsification means breakdown of the fat into smaller micelles. So fat is broken down into smaller particle known as micelles, and that is done with the help of bile juice. So bile juice helps in the breakdown of the fat into micelles. So that is about the bile juice. Intestinal juice is secreted by the intestine, that is mucosa layer of the intestine consisting of or forms goblet cells and brush border cells. So this goblet cell secretes mucus, and the secretion of the brush border cells along with the mucus of the goblet cells forms a succus entericus or intestinal juice. So intestinal juice is formed by the secretion of the goblet cell, that is mucus, and secretion of the brush border cells of the mucosa of the small intestine, and the secretion is known as succus entericus or intestinal secretion. And this intestinal juice consisting of different types of enzyme, that is disaccharidases, as the name indicate, it helps in the digestion of disaccharide, that is carbohydrate. Then dipeptidases, as the name indicate, it helps in the digestion of peptide, means protein, so it helps in the digestion of dipeptides. Lipases helps in the digestion of lipids. Nucleosidases helps in the digestion of nucleosides. So these are the different types of enzyme that is present in succus entericus or intestinal juice. So all these intestinal juice, pancreatic juice and bile juice is secreted into the duodenum, and it got thoroughly mixed with the food particle, and it helps in the digestion of the food particle in the duodenum. Now we'll see what are the chemical reaction that takes place in the duodenum during the digestion. First one, proteins, peptones and the proteoses from the chyme upon entering into the duodenum, it is acted upon by trypsin, chymotrypsin and carboxypeptidase and convert them into simpler form, that is dipeptide. Similarly, fats are acted upon by the lipases and convert them into diglycerides and monoglyceride. Starch is acted upon by amylase, that is pancreatic amylase, and convert it into disaccharides. Nucleic acids are acted upon by nucleases and convert them into nucleotides and nucleosides. So these are the reaction that takes place in the duodenum. In the next step, these end products are acted upon by the enzymes of the succus entericus or the intestinal juice. So, all these end products are acted upon by the enzymes of the intestinal juice and further convert it into simple soluble form. And this reaction takes place near the wall of the small intestine so that simple soluble substance which are formed is easily absorbed into the bloodstream. So we will see that reaction. First one is diglycerides and monoglycerides which is produced in this step is acted upon further by the lipases of the succus entericus and convert it into fatty acid and glycerol. Dipeptides are acted upon by dipeptidases and convert it into amino acids. Disaccharides are acted upon by disaccharadases and convert it into monosaccharides. For example, maltose is acted upon by maltases and convert them into glucose and glucose, which is a simple soluble form. Similarly, nucleotides acted upon by nucleotidases and convert them into nucleosides. Nucleosides acted upon by nucleosidases and convert them into sugar and bases. So all these are the simple soluble form which are formed. Later it will be absorbed directly into the bloodstream, and all these reaction takes place only in the duodenum. So after completion of this digestion or all these process, the food enters the jejunum and ileum from the duodenum. So after completion of all the digestion process, the food from the duodenum enters jejunum and ileum. So there is no digestion process takes place in the jejunum and ileum. Then what happens in the jejunum and ileum? Only the absorption of a simple soluble form takes place in the jejunum and the ileum. So all the simple soluble forms will be absorbed directly into the bloodstream in the jejunum and the ileum. And whatever the undigested or unsoluble waste will be carried into the large intestine. So from the ileum, the waste enters the cecum part through the ileocecal valve, and this valve prevents the backflow of waste from cecum to ileum part. So now the waste is present in the cecum and it enters all along the length of the large intestine, and even in the large intestine there is no significant digestive activity. Then what happens in the large intestine? In the large intestine there is the absorption of water, minerals and the certain drug takes place. Apart from that, large intestine also produces mucus which helps in the attachment of waste particle together, and it helps in the lubrication so that easy passage of waste takes place all through the large intestine, and it will be temporarily stored in the rectum. So that is feces, that is waste, will be stored temporarily in the rectum till defecation. So that is how the complex food particle that is ingested into the oral cavity or the buccal cavity is converted into simple soluble form, and whatever the undigested food particle forms a feces in the digestive system, which is nothing but the digestion. I hope this video is useful. If you like this video, please hit the like button and subscribe to my channel. Thank you.